This video demonstrates a procedure for mounting Drosophila embryos to image hemocytes, their embryonic macrophages. Embryos are laid by flies on apple juice agar plates in a laying cage overnight. The next day, the agar plate is removed and the embryos are washed from the agar plate to a cell strainer. Following additional washes to remove debris, the cell strainer containing embryos is submerged in bleach to decorionate the embryos. After washing off the bleach, the embryos are transferred to a droplet of water. The droplet of water is then aspirated and the embryos are covered in halocarbon oil. Appropriately staged embryos with visible hemocytes are transferred to a Petri per membrane between two affixed cover slips and carefully aligned in oil. A third cover slip is then placed over the embryos and glued into place with nail varnish. The motility of hemocytes within the embryo can now be imaged through the uppermost cover slip on a microscope.
Hi, I'm Jennifer Zanet from Brian Stramer Laboratory at the Randall Division of Cell and Molecular Biophysics at King's College London.
And I'm Iwan Evans from the Wood Lab in the Department of Biology and Biochemistry at the University of Bath.
Today we will show you a procedure for mounting Drosophila embryo to live image the hemocyte, the embryonic macrophages of this organism.
We use this procedure to study the developmental dispersal and wound responses of these important immune cells and the cytoskeletal machinery they utilise to carry out these processes.
So let's get started.
Begin this procedure by obtaining appropriate Drosophila lines containing hemocyte-specific GAL4 drivers and genetically encoded fluorescent reporters under UAS control. Here, serpent GAL4 is used to drive expression of a fluorescent red nuclear marker.